Welcome to this FastFacts presentation from Cell and Gene Therapy Insights and BIA Separations, now part of SARTORILS. I'm Charlotte Barker, an editor at BioInsights, and today we'll be learning how an adenovirus vector manufacturing platform using CIMmultus QA can assure the supply of safe vaccines. Our presenter today is Hannah Hugh, Project Manager in Process Development for Viral Vectors and Vaccines at BIA. If you would like to ask Hannah a question about her presentation, simply use the Ask a Question box to the right of the viewing pane. And with that, I'll hand over to Hannah to introduce herself and kick off today's presentation.
Hello, my name is Hannah Hugh and I'm Project Manager in Process Development for Viral Vectors and Vaccines at BIA Separations, a SARTORILS company. During a 10-year period of working at BIA Separations, I got experience in downstream purification and analytics of demand in biologics. I managed several projects for clients in clinical phases, mainly in field of gene therapy using adeno-associated virus, as well as in vaccine area like adenovirus platform. I studied biology in University of Ljubljana, Slovenia. Today, I'm going to present you our adenovirus vector manufacturing platform using CIMmultus QA. We at BIA Separations SARTORILS focus on developing products as well as solutions for purification of therapeutics, mostly used in the field of cell and gene therapy and vaccines. We manufacture market-leading CIM-monolithic chromatographic columns for purification and analysis of large biomolecules such as viruses, plasmids, and mRNA. Our abounding portfolio is offering solutions to each challenge. Thanks to the increased production capacity, we drastically improved our stock management system and can provide faster turnaround times. Most of our products are ready from stock, meaning that our lead times will not stand in your way when meeting the deadline. Besides manufacturing CIM product, BIA also offers process and analytical development service known as Cornerstone Biomanufacturing Development Service, available for different biomolecules. BIA's technology for manufacturing scale purification is already used in the production of the first commercialized advanced therapeutics and has also a keen presence with novel drug candidates in the clinical pipeline. Today, I want to focus on the solutions that we are offering for adenovirus purification and analytics. In the past, this viral vector had a leading role in gene therapy, but it was later replaced with AAV vector due to some safety concerns. Recently, adeno has regained the attention and is used as a COVID vaccine delivery hub. The everlasting COVID-19 crisis is dramatically displaying the need to expedite vaccine development to mitigate global uncertainties and fear. Consequently, this situation and its dimensions call for the most innovative, flexible, and productive bioprocessing approaches and tools available. And here is where monolithic technology outshines the rest. Because we are all aware of the importance of this viral vector, we have designed plans in a way that we can offer adeno customers immediate support, either by offering columns or by developing methods for them. Allow me to present our second generation adenoviral vectors purification process based on monolith chromatography using CIMmultus QA as the key purification step, as well as accompanying analytical tools. The novel industrial process secures better purity at higher yields, and its robustness was verified using different upstream materials. Finally, higher capacity of the columns used in this process overcomes the raw material supply bottlenecks. Simplified purification of adenovirus usually consists of typical downstream steps listed here, and our case was not exception. We started with cell pellet diluted in buffer and performed lysis by adding sodium chloride to the final concentration of 0.5 molar and 20 to 0.25%. After short incubation, kryptonase, a salt-tolerant nuclease, was added and lysate was incubated for 3 hours while gently mixing. After that, material was clarified with coarse and fine filtration, and then TFF was implemented for diafiltration. Following step was chromatography using anion exchange CIMmultus QA column, where adenovirus was purified in bind elute mode. After capture step, polishing step of ultrafiltration and diafiltration or another chromatography step can be used to further purify or to concentrate the virus as well exchange to formulation buffer. Capture step using CIMmultus QA column was performed on ActaPure LC system. Firstly, pH of retentate was adjusted, followed by another fine filtration. Filtration step before loading seems unnecessary, but it's extremely important in order to remove aggregates formed during TFF. Sample was then loaded on the column and eluted in linear gradient. All unbounded components flew through the column, and in elution we only have adenovirus peak, with low level of impurities before it. Presence of virus in main peak is indicated also by 260-180 ratio. Fractions collected during chromatography were then analyzed using our FastFacts HPLC system with analytical CIMac adeno column. Developed analytical method allow us to track adenovirus through the process and also to follow the impurities removal from the beginning of the process to the end. Additional feature of such HPLC analytics is multi-angle light scattering detector, and it serves as a orthogonal quantification method. Another very important tool we are exploiting in this analytical method is also usage of picogreen staining. Since picogreen binds to DNA, it also helps us to follow the removal of chromatin and DNA residuals known to cause side effects at vaccines. On this slide, we are looking at analytical chromatograms of in-process fractions which disclose a lot of important information. On chromatogram of lysate, we can see high level of well-separated impurities which were then reduced during clarification, TFF, and filtration step. Green arrow here is indicating high flow-through signal which corresponds mostly to the GFP protein in the sample, fluorescing at similar wavelength as picogreen. With TFF, smaller proteins and degraded DNA residuals were removed, as well buffer was exchanged to loading conditions. And then finally, when material is eluted from CIMmultus QA, elution is pure and also complex-free. Additionally, if needed, polishing step can be implemented. This is another TFF or another chromatography step for concentration and or buffer exchange. To sum up, results presented confirms CIMmultus QA to be a very effective tool for adenovirus vector purification. Due to high operational flow rates, these columns enable virus of high concentration and outstanding purity in very short time. Besides, capacity and separation performance is not compromised, and due to laminar flow rate through the channels, shear stress is eliminated, therefore not affecting virus integrity. Furthermore, CIMmultus QA columns provide higher yields at lower manufacturing costs. As well, they represent an alternative for vaccine manufacturing constantly in lack of material supply. Accompanying HPLC analytics using CIMac adenocolumn is a reliable method offering rapid in-process insight for upstream and downstream applications. Thank you for your attention. For more information, feel free to contact us.
Thank you, Hannah, for a great presentation, and thank you to our audience for watching today. If you'd like to download a poster version of today's talk summarising the main points, you can find that below the video. We hope you'll join us again soon for more Fast Facts.